I'm not saying that I've got all the answers.
Have you ever wondered why fat parents have fat children? Or why Chinese parents have Chinese children? It's no coincidence. It's because of DNA. Hi, I'm Troy McClure.
It's kind of funny. I don't know why him taking his helmet off would cause the alarm to go.
You may remember me from such medical films as Alice Doesn't Live Anymore and Mommy, What's Wrong with That Man's Face?
But Mr. McClure, what is DNA?
What the? Oh, hi, Billy. Let's find out together. DNA is God's recipe for making you. You take a dash of Dad, a pinch of Mom, then we bake for 9 months and... that's good, Billy.
Mr. McClure, what does DNA stand for?
So,
Back around the year 2000, there was this thing that was kind of a big deal in genetics, the human genome.
So that Simpson cartoon was kind of obviously a parody on a little bit of virology with the taking off of the bubble suit, and also a parody on the Human Genome Project. Now, what I've been trying to make the argument and getting better at making it, more precise at making it, better at describing the idea, is that in the 50s, after the splitting of the atom and the end of World War II, there were a lot of physicists with a lot of things, not a lot of things to do, and they went into biology. And the idea is that after the discovery of DNA, everybody in the 50s thought that in 20 years we would be engineering babies. Now we're not doing that yet. But in the 50s, it seemed like a reasonable proposition to say that in 20 years, given the speed that we were moving, given the amount of people that we were able to incinerate in Japan, given the amount of people that they were able to incinerate in Germany, it seemed likely that the technology of, engineering babies was just around the corner. I mean, imagine all the experiments they did on all the Jews that they had in those camps. I mean, they must know a lot, right? And so all of these assumptions were being made by people like Joshua Lederberg and written about by people like Isaac Asimov. And so the question only becomes, the people that were writing about the worst case scenario, the people that were projecting the best case scenarios, people that were projecting the future technologies and the future abilities, including the ability to control reproduction or control civil opinion or to control the genetic, destiny of children. All of this stuff was presumed, just like, I don't know, maybe even time travel at one point in time seemed like a science fiction that only required an amount of time in order for it to come true. And this sort of, this is what the foundation of science fiction is. Now, that's all well and good if you're just buying books and selling books and making movies. But if you are administrating a bureaucracy, if you are managing a national security state where nuclear weapons and biological weapons and terrorism are all threats to the entire apparatus, then the stories that you tell about worst case scenario are the stories, the myths, or the realities that motivate ultimately what direction this apparatus is going to move, what priorities the apparatus is going to have. And the scarier the stories, the more likely it is that these will be made into priorities. Now, you can tell all the scary stories you want about aliens and the need for, interplanetary weapons, but because the nature of that threat is very difficult to sell, then that's not the threat that we are being sold. However, the biological threat, the molecular biological threat of viruses, of disease, of biological weapons with very little specificity as to what a biological weapon is, these concepts have been pushed for decades now, and some of those concepts are based on conjecture that with the new ideas and data and thought and experiments that we've had and done, we now know that that conjecture is ridiculous. And now the question becomes, what parts of, if any, the Human Genome Project were projections that were either purposefully scripted dumb or have had to be scripted dumb in order to hide a lack of progress. Now remember, what I'm trying to suggest to you is that these worst case scenarios, while they may have been plausible at one point in time, no longer are plausible. And that the reason why we can see that is because there are a few narrative glitches at this point in the mythologies that we have been coerced into believing. And these narrative glitches are permanent scars that reveal the illusion of the theater and the illusion of the cast and the illusion of there not being a single script but a bunch of individual actors, which is just ridiculous. And so now the idea would be, what kind of video would be produced during a time when, again, the Human Genome Project is ongoing, not over, and our sovereignty is being inverted to experimental animal status so that they can take all of our medical data and all of our genetic data that they can glean from all of the testing that they've done and all the testing they continue to do, all the remnants they continue to generate. And why it is that people aren't more aware of this. Because when I was a high school teacher, when I was a college student, we were in the midst of telling everybody that these breakthroughs were being made, that we were, again, a decade away from real progress in understanding how, like, single gene diseases like sickle cell anemia could be cured. But if it turns out that in 2025 we can't cure sickle cell anemia like we thought we were going to cure sickle cell anemia, that would be pretty extraordinary, right? If we can't cure Jesse Gelsinger's of today in a way that we thought we could cure Jesse Gelsinger's in the year 2000, like, 25 years ago, then some progress, or no progress rather, has been made. And that lack of scientific progress needs to be disguised. They need to kick the can down the road so it seems like, yeah, it's still right over there. We told you it was right over the hill. You can just barely see it. It's shining in the sun just over the top of the hill. That's where the can is. Keep running. And so now, one of the things that I want you to remind you of is that one of the only ways that they've been able to sell the Human Genome Project as successful is the ever exponentially decreasing amount it costs to do a given sequencing job. So it used to be very expensive to sequence a human. Now it's much cheaper. It used to be very expensive to sequence DNA of a bacteria. Now it's much cheaper. And that's the main story that they have to tell you back then and all the way to now. And so now we got to kick the can again because, again, the biological sort of theater of COVID-19, I don't think was successful. I don't think it was as successful as they wanted it to be. There's too much fractured opinion. And so it's going to be tricky to get this ship back on course. The DNA contamination with Kevin McKernan is not going to do it. And so it's just curious. I just want you to listen to this. I'm going to stop it a few times. And then just always keep in mind that this is a current video just made a couple weeks ago. And more importantly, keep in mind that this woman is talking about the Human Genome Project, the International Human Genome Project, where Francis Collins was the head of it, and Kevin McKernan was one team leader, a research and development director of one team at the Human Genome Project, and was at the announcement, has met Bill Clinton, has taken pictures with these people, was on Geraldo Rivera to announce that the Human Genome Project had been completed, has spun off companies, and is now purporting to be the lead dissident whistleblower on an intergenerational genetic disaster because there was SV40 DNA in the COVID vaccines. This man is a fraud. He belongs in jail, except that he's a national security actor, probably not acting for private interests, but acting for the U.S. government. If he's acting for private interests, then indeed he is a traitor and a criminal. The same could be said for Robert Malone. If he's acting on behalf of foreign governments or private interests rather than what he claims to be as a volunteer ACIP director and, emu farmer, then he is also a criminal. And the same can be said for Pierre Corey, for Peter McCullough, for Jessica Rose. Because these people might also be scripted to make it possible for somebody like Senator Ron Johnson to hold hearings in the Senate and have plausible deniability about what's being said. And, just be like, well, all these people were in my meeting and they said all these things. Am I supposed to not believe all these people? And so that's what gets created. The same thing at Children's Health Defense. A bunch of lawyers that are surrounded by a bunch of scripted actors like Mary Holland and Brian Hooker and Meryl Nass. And so a lot of those lawyers don't know any better because they're not biologists. They know to stay in their lane. They don't have an autistic kid. They know to stay in their lane, and so they do. And so it is an extraordinary place where we are, where the Health and Human Services Secretary, the head of the NIH, the head of the FDA, and the head of CBER are all scripted actors that are being more or less puppeted by Kevin McKernan and Robert Malone, and I guess people like them or behind them or with them. And so this is a real video that's supposed to be like not that big of a skeptical video. It's not supposed to be that shocking, but what you should hear is that what she admits throughout this video is that the predictions that were made 25 years ago did not come true. We're not engineering babies. We're not curing genetic diseases. We're not doing any of this shit yet. But when they announced it, they said they were going to be doing it. And understand that that happened in the 70s too. And it happened in the 60s too. And so we are now here, really 60 years later, and we still can't figure it out. And the only reason why we still can't figure it out is because there are scripted actors making sure we never figure it out. She can't figure it out because she's too busy reading.
Genome Project. Its goal was to decipher and publish the entire human genome for the first time ever. And people, including some scientists and politicians, really hyped it up. US President Bill Clinton claimed it would revolutionize care of almost every disease, for example. One kind of sky-high prediction even said that by 2016, we'd all carry around our own personal genomes on a card in our wallets next to our driver's licenses, and that doctors would be able to prescribe specific gene therapies to anyone who needed them.
Wow, specific gene therapies for anybody who needed them. It's funny because the same argument has been made for 25 years. Peggy Hamburg was making that argument. I mean, Kevin McKernan is making that argument about specific strains of weed for specific people, making specific cannabinoid combinations for specific genetic backgrounds. The anti-vax movers like James Lyons Weiler is making the argument that it's genetic backgrounds that are susceptible to vaccine damage, and some people are just invulnerable. Don't you hear it? She's just reading. She doesn't know how she's being used. She's not sophisticated enough to understand that the Human Genome Project is an elaborate military operation on the scale of 30 or 40 years. It's an international collaboration that's ongoing.
You look back at those kinds of news stories, it's clear that the potential of the Human Genome Project was overpromised. In this episode, we'll talk about what the Human Genome Project was and why so much of the hype that was promised didn't come to pass. But also, how it still kind of revolutionized science anyway.
It kind of revolutionized science anyway. I wonder how it will have done that. Probably by making it cheaper to sequence shit.
I don't know about you, but I do not have my genome in my wallet. Though, to be fair, the Human Genome Project was, in many ways, a success. The project was an international collaboration that ran from 1990 to 2003 to the tune of $2.7 billion. They did end up sequencing the genome, or just about all of it anyways, which was an incredible accomplishment. But as the years rolled on, it became obvious that the promise to revolutionize how we'd approach every disease just hadn't panned out.
And why is that? The reason why is because they need every genome available. Well, we finally successfully played a game of Go. Now all we have to do is play 1 billion more games, and then we will know, we'll have a machine learning algorithm that will be able to beat the game Go. The same thing they did with chess will be done with Go, will be done with the Human Genome Project, and you need as many games as possible. The Human Genome Project was about assessing the size of the problem. It was about landmarks and making the attempt to do it. And then instituting a worst case scenario projection and a best case scenario projection that would allow the focusing of resources on these technologies. So at the end of the Human Genome Project, they understood the depth of the problem, the difficulty of the problem, the kind of thing that they wanted to accomplish, how hard it was to accomplish with a single sample, a single background, and then trying to move forward on how they might do it with a bunch of people, like all the people. Now, I'm not saying they can do that now. I don't know if they can do that now. What I'm trying to suggest to you is that the vast majority of people involved in this are not sophisticated enough to be asking questions on that level. We've got to work from the level of the illusion on some levels. We can't just throw it all out and assume that it's all there. I mean, if you start assuming that there's no DNA, and it turns out that there's no DNA, well, then we should find that anyway. I really believe that to be true. If there's no DNA and DNA is a myth, then at some point in time, with diligent work, we're going to come to that conclusion, just like we came to the conclusion that RNA cannot pandemic after diligent work, that animal viruses have to be, there's something, corners that have been cut since they stopped talking about phages and started talking about animal viruses with David Baltimore's scheme of enzymes. There's something that happened there that just doesn't jive with logic and intellectual reason and the frickin' book timeline. I'm just buying books from every decade, every 5-year period to try and see how people's ideas changed, and I can't find an inflection point where suddenly nobody gave a shit about bacteria and phages anymore. I just find a bunch of people starting on animal viruses. It's really bizarre. And so here we go.
There was no magic bullet for all of our ills hidden in our genes. Why did some advocates promise this then? Some of it was no doubt hype or excitement.
So what's weird already is that she can't feel the cognitive dissonance in her own script. So she says that they promised something, but they didn't manage. There were no secret markers or whatever. It's almost as if she is on a script that's so dumb. I mean, because again, if you say that it's almost too hard to explain how dumb this is, but it is very dumb. But she's just reading a dumb script.
Even scientists and science communicators can get caught up. But there was also some reason to buy in.
See, even science communicators like me, I'm not a science communicator. I'm a frickin' biology coach. You need to do the work. You are the person that needs to do the work. Every responsible adult is responsible for learning what's up. Because it's reality and you live in it.
A couple of papers in the late 80s claimed to find single gene causes for some pretty big medical issues. That included mental health conditions like bipolar disorder, schizophrenia, and alcohol use disorder.
Those were not single gene issues. They were GWAS studies that a couple genes showed up very brightly on. Those are the stupidest thing ever.
These conditions really did come down to just a single gene. All we needed to do to treat them was to tweak that one gene, right? Unfortunately, many of those papers later came under scrutiny, and their findings couldn't be replicated. The truth is, most diseases are not caused by a single mutated gene. A lot of diseases, especially the big common problems like diabetes or the aforementioned mental health conditions, are influenced by genetics. But they're the result of a lot of tiny nudges from many different genes, not just one big mutation.
That's called development and homeostasis, like a pattern integrity with a trajectory across time.
Today, we know that dozens or even hundreds of genes may affect your risk of developing diabetes, for instance. This means that those genes do not lend themselves to a magic bullet cure, because just changing one of them probably won't have any observable effect. Worse, it may have unexpected side effects, especially when you consider that a person's environment and life history also play a role in their risk for diabetes. And scientists and doctors, well, they kind of knew this was the case even back in the 90s. So you've probably noticed doctors don't routinely screen your DNA. Now, to be fair, there are some conditions, what we'd call Mendelian conditions, since they follow Gregor Mendel's basic rules for inheritance, where a single mutation is enough to matter. Diseases like Huntington's or sickle cell are caused by mutations in a single gene. There are certain cancer genes, like the BRCAs, where certain mutations make it much, much more likely for you to get breast cancer. And there are some medicines, like the blood thinner warfarin, that work significantly better or worse in people with certain mutations. Also, and this is cool, if you already have certain cancers, doctors will sometimes do genome sequencing on samples of the cancer cells.
Now remember, that we were making this argument the other day, I think, but experts today are not experts like they used to be. They're not experts like they should be. Science communicators are even worse. Anybody that calls themselves a science communicator is already admitting that they don't know what they're talking about. They read scripts. They memorize scripts. They can improvise within a script, but they have no understanding of the biological principles on which those scripts are purported to be based because they can't explain them. Simply put, anybody who's an expert should be able to explain their expertise for a dum-dum. Otherwise, they're not an expert. And that's the extraordinary thing that we've allowed these people to get away with. We've allowed virologists to get away with it. A lot of these public health immunologists and epidemiologists get away with it because, again, on a podcast, with a podcast actor on a script, you don't have a real dialogue. There is no logical progression. There are no interrogative questions. This is all bullshit. And she cannot talk about what she's talking about. She can only read this script, and she reads it in such a way, they cut it in such a way so that when she stumbles, she just starts again.
Cells themselves, since screening for specific mutations can help your care team spot weak points and choose treatments. So there are times genome sequencing really does make a big difference in medicine. But it's worth noting that in all these cases, scientists already know specific things to look out for. Doctors don't really just blindly sequence an entire person's DNA and then go looking for oddities. Because, to be honest, knowing that a DNA mutation is there doesn't really help you figure out what that mutation does. Like, let's say you have—
Because a lot of them are actually silent mutations. Mutations that change the codon but don't change the amino acid can cause disease, apparently. And we have no idea why.
A single nucleotide difference between two people's DNA, even if you know it's part of a gene, which we'll come back to.
So she's already implying that we could look at your genome and my genome and be relatively sure that there's a single nucleotide difference between the two of them. And I really am skeptical that we have that level of fidelity. I really am. It's not saying I don't believe there's DNA, but I am skeptical that sequencing technology has that level of fidelity.
It doesn't necessarily tell you what the gene does or whether the mutation matters. At this point, it's still all TAGC gobbledygook. It's possible that a scientist could look at that and make a guess about what that change would do. But unlike computer code, the genetic code doesn't contain helpful little annotations to say, this part does this. You can't tell what the genome does by reading it. You have to sit down and do experiments to see what a change functionally does.
So with billions of bases and so many non-coding gene sequences, even if we give them most of the basis for this myth, it doesn't make any sense. She's telling you that the Genome Project could not have done what they said they could have done. And she's telling you it in 2025, 25 years after. And in 2000, when they were doing it, they already knew it. Kevin McKernan already knew it. They all understood where they were trying to go, and the mindset that they were trying to change. That's why Kevin McKernan himself was on Geraldo Rivera. And that's why Kevin McKernan was in front of us in 2020 talking about how the FDA is in the way. And that's why the pandemic is running rampant on North America, because the FDA is in the way. The EUAs require 10 pages of paperwork. When anybody could test for this coronavirus, we could be doing it on benches. This man, Kevin McKernan, is one of the biggest frauds in America today. Like Robert F. Kennedy Jr., like Meryl Nass, like Mary Holland, like Brian Hooker, and like Robert F. Kennedy Jr., like Jayanta Bhattacharya, like Vinayak Prasad, and like Marty McCary. They are all frauds on a script provided to them. They basically podcasted their way to the highest levels of American government. We are being sabotaged right now. And on the internet, there are videos which actually are there specifically to kick this can down the road to make you not realize how much we've been bullshitted. Hoodwinked.
It's much easier to start with a known disease, like diabetes, and then work backwards, looking for mutations in proteins we already know are important, or comparing and contrasting a lot of sequences from people with and without the disease. So that's what we mean when we say people back around the year 2000 probably overpromised things about the Human Genome Project. The project did succeed.
So what do you think they overpromised after the Manhattan Project was over and these people went into the genetics of bacteria and phages? Well, they promised everything. They promised the ability to take over the evolution of the human species. And that is the promise they still believe in today. All these other little stories and goals and whatever, they're all bullshit. They still believe, like they did in the 50s, that man is destined to take control the evolution of the species, that we are destined, that God put us here to do that. We're not gardeners. We're builders. And I think that's wrong. I think humans were put on earth to be the gardeners. I think if we put our minds to it in the way that we should do it, if we put our kids' minds to it in the way that we should do it, all of us would be living in an abundance. And the vast majority of us would spend most of our time with animals and four-wheelers and sports. But of course, that's not what human culture has been bent toward. As Buckminster Fuller said in that 1967 video that we watched the other day, it's consumers are really the thing that allows large state projects to happen. You need a prosperous living population in order to create the amount of money and resources and momentum to do projects as big as like an aircraft carrier that's powered by a nuclear reactor. You can't get enough, private interest in that to make that happen. You need a juggernaut of economic and social will that can only be achieved by a national system, a nation. And so a nation with more people is going to be able to accomplish more than a nation with few people and comparatively fewer resources. And so a lot of the projects that have happened in America and a lot of the resources that have been put into those projects are because of our population being so prosperous and our country having so many resources at its fingertips. China is another example of how a population can be focused on certain goals and then resources are harnessed and put to work. And so India is now the largest potential energy source of that kind on Earth. And these people understand that. And so as we move forward in human history, and the dominant population that will fuel the big projects and fuel the big endeavors is shifting away from the United States and shifting to other countries, like maybe China for a little while, but India for sure. And this shift is real, and the people who are playing this game for real are playing this game with the real, the real numbers. The real numbers are that Americans' population is going to collapse. Western Europe's population is collapsing, and it looks like immigrants are being put in a place to kind of replace that population. It's not a conspiracy theory, it's math. If my brother and his wife have decided not to have any kids because it's selfish for the planet, and their next door neighbors are having children and they're from Pakistan, well, then that's what you get. There is a responsibility that every adult human has, and that is to seek to contribute to the next generation in some way, to plant trees under whose shade you will not shelter. Those trees could be children. Those trees could be your brother's children or your sister's children. They could be children of neighbors that you deem worthy of your time. But if you're not contributing to the next generation and you're not contributing by planting trees under whose shade you will never shelter, then you are an irresponsible, useless human being. I think that's an interesting take that's very similar to what Bill Cooper said in the beginning of his book, Behold a Pale Horse. You just got to think on the right time scales, and you got to stop thinking on the time scale of social media. Start realizing that if you don't contribute to the next generation, someone else will. And there is a certain math to be done. If we don't have kids, there won't be anyone alive in 100 years. You realize that, right? If everybody just stopped having kids right now, all humans would be gone in less than 100 years. It's a guaranteed certainty. So it's a pretty huge decision to make, to not have children. You better be pretty certain about the circumstances that are generating that decision. And yet most people are still fooling around, memorizing scripts, memorizing mythologies, and then arguing about them without ever developing the expertise to question them. And that's how academia fails us. That's how our schools fail us. That's how our teachers fail us. Because if we have math teachers that can't teach math, but can only correct tests, then our kids are in trouble. You know that in your heart. Well, we right now have biology teachers and we have medical school teachers that can only correct tests and tell you what's going to be on them. They can't actually explain why that content is on the test. They can't actually explain why we've come to believe that that content is correct, that those hypotheses are worth investigating, that this data indicate. They have none of that in medical education and none of that, for the most part, in academic biology anymore. Even though in math, it's very hard. In physics, that's very hard. In engineering, it's almost impossible. But in biology and medicine, they have done it. And now we have a comic level of science communication. There's no engineering communication, people. There's just engineers that know what they're talking about and people that are full of shit. There's no There's no carpentry communicators that bridge the gap between the impenetrable art of carpentry and, using the drawers and assembling an IKEA product. But somehow or another, we have to have science communicators because the scientists are so smart that they can't explain what they do. That's where you can see they're lying. That's how this script breaks.
Successfully sequenced the genome, but saying it was going to revolutionize how we treat nearly every disease was skipping a lot of the steps in the middle. And there was something else they couldn't really have known just yet. Here is...
I need to step in for a minute with a huge shout out to the company that supported this video and so many other SciShow videos in the past. Brilliant. Brilliant and SciShow have a lot in common.
Now what you see here, right, of course, in a company like Brilliant is a company that's going to try and make money by making, web pages that you pay for. And they can say that they're teaching you things with these graphics, and maybe they are teaching you some things, but the most that they are doing is wasting your time because they don't have any priorities about what you should learn. They don't have any priorities about what's wrong in society right now. What are the things that, if everybody understood, would be the best? They don't have anything like that at all. What they're doing actually on this website often is teaching you how AI works or how large language models work, kinda, and getting you to believe again than what you see when you're told on the internet's going on is just an advanced version of that, and then they teach you the little bits.
What scientists didn't know was what all those letters in the genome actually do. Now stop me if you've heard this one before, but it's a really common metaphor to refer to DNA as a kind of writing and the genome as a book. Metaphors are good. They're a good starting point our brains can hold on to as we learn something new and complicated. But metaphors are also at least a little bit fiction, and it turns out the real genome is much weirder than a book. The human genome is about 3 billion letters long. At the time, it was assumed that most of those letters would code for protein.
At the time, it was assumed that most of those letters would code for proteins. It's interesting, she's got it all right. She's got it all right, except she can't figure out why it is that they would be still lying about it then.
As a quick reminder, DNA gets read out or transcribed into messenger RNA, which gets processed and then further read out or translated into protein. And proteins are what do much of the work of being alive. So it was not actually a stupid assumption to think most of the genome would code for protein, but oh boy.
Well, it is actually, because if you think about it, that would be like saying that all of the words in a book are verbs. All of the sentences in a book are declarative. It's just absurd. It's absurd, but she's reading a script, so as she goes through them, hits the many absurd notes of Biology 101, she's just memorized it.
It does not. The sequence of the genome revealed that the vast majority of our DNA, something like 98 or 99%, doesn't code for proteins. It was as if most of the pages of the book were filled with nonsense, what some scientists called dark matter, or more pejoratively, junk. Firstly, for instance,
dark genetic matter is what Nathan Wolfe called all of the signals that they were working on using metagenomic sequencing to find viruses in the wild.
There's DNA sequences that affect how genes are expressed without actually being genes themselves. Promoters, for instance, are stretches of DNA just ahead of the protein coding part of a gene. They are where a bunch of cellular machinery will come sit.
Now, this can be true in the context of a bacteria and a bacterial circular plasmid. There are sequences that you can add to other sequences to make those sequences get copied, and I think that a lot of those experiments have been pretty thoroughly done. But the assumption here of Biology 101 is that all the molecular observations that we've made in a bacteria are assuredly at work in our own cells in exactly the same way, doing exactly the same thing. It's just a little more complicated in our cells. And of course, this is also based on the idea that we are descended from bacteria, which is, of course, another really dumb assumption to make just because, I guess evolution because DNA. Here we are again. This is just cartoon enchantment, implying a level of fidelity of understanding that we don't have.
It down to read that gene out, a little like markings on a runway.
Just because you have a line that says RNA polymerase and a line that says repressor and a line that says promoter or a word doesn't mean that those things are real. I could go outside right now and put a bunch of labels on my car, and a mechanic or even a person with common sense would know that a lot of those labels were wrong, right? If I called the alternator a re-encabulator, come on.
There are also enhancers, repressors, and silencers, which are sequences of DNA that either increase or decrease expression of a gene. There are also parts of the DNA sequence that are transcribed into mRNA, but for some reason do not end up in the protein. Introns are DNA sequences that get cut out of the mRNA before it gets translated into a protein.
And we don't know what any of them do, although they must do something, and we can't take them out one by one and see what happens, so we're kind of screwed.
Sounds weird, but it's actually a flexible system that can lead to different versions of the same protein by chopping the RNA up in different ways. Sometimes DNA codes for RNA that has a specific job other than becoming a protein. For instance, the ribosomes that translate mRNA into protein are, ironically themselves, mostly made out of RNA.
So the most important process in life is the translation of RNA to protein. At least you might make that argument. It's the process by which proteins are folded, and if you misfold a protein, you can get a prion, and then you're dead. So it's a pretty important point in biology, you know, that point right between RNA to protein that they often just gloss over. It's like the machine. And we don't think about the differences between my ribosomes and yours. We don't think about the differences between the ribosomes in one tissue versus another. We don't think about the changes in our ribosomes over time. We have a guy named Debunk the Funk who did a PhD on the subunits of ribosomes, I guess because he thought they were cool, and now he's a guy who works for a testing company and fights the RFK anti-vaxxer people. Because the most interesting machine on Earth wasn't that interesting. I mean, this is a joke to me at this point.
There are also bits of DNA that seem to be more like cellular scaffolding or structure than instructions, like telomeres, which are long sequences of repetitive DNA that kind of...
I mean, ribosomes universal? I don't know how much we know about the sequence of ribosomes and how universal they are. I do know that I'm told that there are ribosomes inside of a frickin' mitochondria.
Cap off the end of a chromosome and keep it from unraveling. And finally, yes, some of it does seem to actually be junk. There are things known as pseudogenes, which seems to essentially be broken genes that no longer function. Even the 3D architecture of the cell's nucleus can affect gene expression. Like very long noodles in the worst plate of spaghetti ever, chromosomes have to be arranged in 3D space in order to work. The enhancers I mentioned earlier loop around from really far away to affect the genes they regulate. In contrast, DNA that's packed up really tightly is harder for cellular machinery to get at, so it's expressed less. And all that's just scratching the surface. There's more weird stuff going on, as well as stuff we don't quite understand yet. The point is that the genome is really complicated, and it's about a lot more than reading letters from beginning to end. Basically, our book is making House of Leaves look straightforward. So here is an alternate metaphor. It's still a bit of fiction, because all metaphors are a bit of fiction, but humor me. The genome may be less like a book and more like a library. There are books with blueprints and people copying down what they see inside. But there is also scaffolding and shelving. There's Dewey Decimal numbers and reference maps, locked up sections and big open promotional display cases, and internal documents and memos. You might be
I'm going to let her finish.
Be able to see a few librarians reshelving things or shuffling them around, and yes, even a few tattered junk books destined for recycling. I think that's a much cooler concept than a book.
And then what she will miss here and can't really explain to you is that you can't walk around in the library, you can't pull books off the shelf. The only thing you can do is pull random pages out of the library. At best, maybe about a page and a half out of any given book. And in order to do that with any fidelity, you have to know the beginning and the end of those passages to a pretty good extent. Otherwise, your primers won't work. And that's all you got. If you want to sequence the whole library, you have to understand the beginning and end of every 8 or 9 chapters, right? Because you have to have primers that are specific for that thing. And if you don't have that, then what you're amplifying can't be proven to be what you... it's all very, very complicated, of course. And so Kevin McKernan will be able to tell you with absolute certainty that Jonathan Cooey is full of shit and that the Human Genome Project is exactly as far as they say it is, because otherwise he wouldn't have a house in Marblehead outside of Boston in the most expensive real estate in that area on the ocean with $10,000 cats.
Combining both the points about diseases and the complexity of our DNA, we arrive here. In sum, knowing the entire sequence of a patient's genome would likely do very little for their care outside of certain situations, and that probably won't ever change much in the future. The big diseases are usually the result of many small nudges rather than one big push, and not all of those nudges lie in the genome. And the genome itself is really complex, so even if you know of a mutation, that doesn't necessarily tell you that much. So that specific promise about being able to sequence everyone's DNA, leading to easy cures to everything, never came to pass. But here comes the twist. Does that mean that the Human Genome Project was a failure for science? No, it does not. All that stuff I just talked about, we were able to learn about because the Human Genome Project provided a scaffold for everyone else to work from. In many ways, the Human Genome Project, like the moon landing, was primarily a technical achievement. The techniques they used paved the way for modern sequencing technologies. Today, what once cost billions of dollars and took 2 years is down to less than $1,000 and can take as little as 5 hours. A scientist can whack a sample down in a fancy sequencer, go have a sandwich, and look after a few other experiments, and come back to the results before it's time to quit for the day. And this proliferation of cheap, reliable, and fast genome sequencing did indeed revolutionize science. The tools we got out of it are used in everything from understanding basic cellular processes to studying evolution to ancient archaeology. What's more, going wide and being able to test the genomes of a lot of different people may deliver some of the benefits that we didn't get from doing it once.
Wow, it may deliver some of the benefits that we didn't get from doing it once. Well, no shit, Sherlock. In fact, I would make the argument that they've always known that, and that the COVID pandemic was designed to allow that, to create the remnant streams necessary to allow it, to create the databases to allow it, to create the AI infrastructure that would allow it, and the, yes, that's correct, the institutional inertia and momentum required to do it. Requires myths that large populations believe so that those resources and their resources can be devoted to it. The big projects, ladies and gentlemen, the big projects.
Because there's power in being able to sequence a lot of people. For one thing, it helped us get a better grasp on the diversity of the human gene pool. The original sequence the Human Genome Project published was a patchwork of just a handful of people. Actually, about 70% of the genome came from one anonymous sample. Today, though, subsequent efforts like the 1,000 Genomes Project...
The 1,000 Genomes Project is a project on which Claire Craig, the co-author of the Corman-Jorston report with Kevin McKernan, the one that objected to the WHO PCR test that Mike Eden was on, she is the head of HEART, a dissident organization in the UK. She is promoted by all the people externally from the United States. She's like the most important person that John Campbell can talk to. And she has the same glasses as Liana Wen and Whitney Webb and Courtney and all these other people. They're all wearing the same glasses now. Even Sasha Alatapova has the same glasses on. The 1,000 Genomes Project is where Claire Craig worked before she was recruited and scripted to kind of complain about PCR as a, fast food versus fancy dining kind of thing. in 2021, the same time when Robert Malone came out on the Brett Weinstein podcast to stand in front of Byram Bridle, who apparently now has a wife who's going to get all of her limbs amputated, but he's still got enough time to Substack about bird flu and ostrich farms. Claire Craig is a part of this genome project that she started talking about right here. If you think for 1 second that this isn't the biggest story in modern human history, then you're not paying attention. And that's nowhere else on the internet but here, right here, nowhere
Project
else.
give us a much better sample size to work with, allowing us to understand not just how one person's genome works, but how genetics affects people all around the world. The Human Genome Project era also gave way to the era of what's known as Genome-Wide Association Studies, or GWAS. In general, this involves rounding up a bunch
But why didn't she explain this in the beginning when she was telling us about those genes for schizophrenia and for depression? Because that's where this came from. It's not that great. In fact, it's pretty dumb. And so she should have probably explained this in order to, give us a little bit better understanding of what she was saying before. But because she's on a script and she's not a very good science communicator, it's just god-awful.
of genomes and looking for patterns or associations between a disease and a bunch of genetic variants. Results from GWAS may translate into discoveries for diabetes, autoimmune disorders, and schizophrenia. That's how we got those hundreds of diabetes genes. The contribution of each one is so small that a pattern only emerges when you're looking at loads and loads of people.
See, and the thing is, loads and loads of people will allow the pattern to emerge, but then you still are under the assumption that everything that you're finding with these kinds of sequences is a legitimate signal, which is a stupid assumption.
There still will probably never be a single magic bullet gene therapy type thing for every disease, but
Don't forget that Robert Malone thought they would be using retroviruses to cure diseases exactly like they describe here was not able. I mean, David Baltimore thought the same thing.
these discoveries can give us a better understanding of how these diseases occur and the best ways to treat them. The Human Genome Project helps scientists at the lab workbench more than it helped patients in the doctor's office. Of course, the scientists at the lab bench do help the people in the doctor's office eventually. It's just more roundabout than we'd hoped. So that's the story. The Human Genome Project was a huge achievement that may have been overhyped, but it was still a huge technical success and is still leading to exciting new things. That said, with the benefit of hindsight, we'll resist making any huge promises about what'll end up in your wallet in the future. As we've learned, genetics can be far more complex than it initially appears. If you liked this episode about why the Human Genome Project was a failure, but still a good idea, you might also like our video about why the moon landings were a failure, but also a good idea. Thanks for watching.
That's funny.